Okay, thank you, Steve. And thank you very much for inviting me to come and give a talk in this seminar series. I have to admit I had a sneaky look on the website a week or so ago and actually listened to one or two of the lectures. So I realise I've got a lot to live up to. Anyway, so as you heard, the focus of my talk today is going to be on the prefrontal circuits underlying anxiety and anhedonia. And we're interested not only in establishing the circuits that underlie the regulation of both positive and negative emotion, but also trying to understand how current therapies used for treating anxiety and depression, actually what those neurobiological mechanisms are. I know from having seen the talks you've already had at this series that you've heard a lot about the problems, the kind of factors that currently are making progress very slow in understanding the etiology of psychiatric disorders as well as their treatments. So I'm not going to dwell too much on that today, but I'm just going to give you my take on it with respect to anxiety and depression, but particularly with respect to depression. And I think there are some sort of three key things to realise. The first is two patients can be diagnosed with depression and have no overlapping symptoms. Even if two patients have the same symptom, the psychological and neurobiological underpinnings of that symptom may well differ, and that's primarily because the clinical symptoms of anxiety and depression have been poorly characterised up until now. And finally, even when treatments do work, we don't actually know how they work, and therefore we can't predict which treatments are going to work best in which patients, which means patients are still being used as guinea pigs for trying to work out which drug is going to be best effective for that particular individual. So for all those reasons, as I know have heard from other speakers in this series, we need better stratification of anxiety and depression, and we need to be able to develop personalised treatment strategies. We have here a relatively simplified schematic of the brain and the various structures in the brain that we know are already associated with emotion. And I was just looking to see if I've got a pointer, electronic pointer anywhere. Is it in here? Ah, brilliant. Thank you. We know subcortically that structures such as the amygdala and the bed nucleus of the stria terminalis are key components of the subcortical emotional system, both in the acquisition and expression of emotion, and that through their projections out to downstream structures in the brainstem, in the hypothalamus, and up into the striatum are involved ultimately in orchestrating the expression of our emotions. And we've gleaned a lot of information about this circuit from studies in rodents. But we have far less of an understanding about the cortical regulation, modulation of this downstream circuitry, in particular the prefrontal cortex shown here in pale blue. We know that some people are more or less likely to go on and develop a psychiatric disorder. And one of the factors that make you more vulnerable to developing anxiety disorder is, in fact, anxiety itself. Anxiety is an adaptive emotional state. It's important for us to be able to avoid negative stimuli in the environment. But if it's chronically engaged, it can become pathological and ultimately go on to lead to a variety of different anxiety disorders or indeed depression. But what makes you or I either low or high anxious as a behavioural trait? Primarily, it's a combination of the genes we were born with and environmental experiences as we were growing up. And together, these interact to ultimately affect the development of these circuits, which ultimately may lead to cognitive and behavioural dysregulation later on in life, and ultimately make us more or less vulnerable to developing a psychiatric disorder. As I said, the most important regulation of this circuitry, the subcortical circuitry, is the overlying cortex, in particular the prefrontal cortex. And that's why I'm wanting to do studies in primates, because by far the most complex cortical structure is found in monkeys and humans. And this can be seen in this next picture, which just shows you how much cortex or how much of the brain is given over to the cortex in rodents right up to humans. In rodents, 28% of the brain is cortex, which is where the highest processing of information is going on in the brain. In humans, it's 80%. And if we turn and look at monkeys, New World monkeys, the marmoset that I work on, and Old World monkeys, the rhesus monkey, you can see that, again, of the brain, 60 or 70% of it is made up of cortex. So that straight away shows you why, within an overall experimental strategy, we need monkeys at the center of it. And if we look specifically at the prefrontal cortex, which we know is very important in regulating emotion, this shows you the prefrontal cortex in humans, in rhesus monkeys and in marmosets. And what you can see overall here is that the structure looks rather similar. Based on overall structural analysis, there are a number of different regions within the human and monkey prefrontal cortex, ranging from areas 9 through 10, 11, 12, 13, and 14, and then anterior cingulate regions 24, 25, and 32. And they're all rather similar across the species. Whereas what you can see is when you turn to the rat, whilst it has a very distinct parcellation, we don't know how these areas map onto the equivalent, if there are equivalent areas, in the primate. For all these reasons, then, I've chosen to work on a primate, and I've chosen to work on the marmoset. And in our lab, we have approximately 4 different experimental strategies. The first is to actually manipulate various structures within the prefrontal cortex and related areas in order to establish cause and effect. Brain imaging has told us a lot about what's going on in patients with various psychiatric disorders, but ultimately we can't establish cause and effect. We can't determine whether particular areas of altered activity in the brain in a human patient actually causes a particular symptom. Even if we look at the brain changes in brain before the disorder comes on, you still can't establish causality. For example, some of the changes you see may in fact be due to compensatory effects. So the brain may be compensating for some of these changes. So in order to address that issue, you have to turn to experimental studies in animals. So that's why in the marmoset we look to see, for example, if specific alterations in activity in one or other of these prefrontal regions can actually cause specific deficits that look like anxiety or anhedonia. Combining with our manipulation studies, we've started to use micropet, in particular fluorodeoxyglucose, which allows us to measure activity in the rest of the brain when we make these manipulations, that will allow us then to see what is the knock-on effects not only of our particular manipulation on behavior, but also what's happened to the rest of the brain as a consequence of that manipulation. Also in the lab, we are starting to study the various traits, behavioral traits that we know make you more vulnerable to developing psychiatric disorder. So we originally now started looking at trait anxiety in marmosets. They show a massive range of differences in their anxiety levels, and by studying them in a breeding colony, we're able to start looking at the neurobiological mechanisms that may underlie how anxious you are as a trait. And then finally, more recently, we started to look at genetic risk factors, and we focused on the serotonin transporter polymorphism, which has been implicated in developing anxiety and depression in humans. And we found that whilst we don't have the exact same polymorphism in marmosets, there is a very similar one that seems to have very similar functional consequences. But what we can do in the marmoset, which of course we can't do in humans, is actually study the underlying neurobiological and, importantly, the molecular and cellular mechanisms that explain how a change in the genetic polymorphism can ultimately have such an impact on behavior, which it does in the marmoset. However, for today's talk, I'm going to focus primarily on these 2 strategies with respect to anhedonia and anxiety. And so I will first tell you about some recent work we've been doing looking at the orbitofrontal and ventrolateral prefrontal regions in the marmoset and their interactions with downstream structures, including the amygdala and the hippocampus, with respect to anxiety and negative bias. And in the second half of my talk, I will focus on anhedonia, the loss of pleasure, and look at areas of the brain in particular caudal subgenial prefrontal cortex, area 25, and its interaction with the dorsal anterior cingulate cortex and the insula. And some of you who were at Professor Etkin's talk a couple of months ago may remember that he actually showed you how important anxiety and anhedonia may well be with respect to biomarkers of depression, but not on their own, but in relationship with altered structural and functional changes in the brain, and how together anxiety and depression and their associated changes in the brain may in fact allow one to separate out different components of depression in different patients. In all the studies I'm going to show you, there are some important principles that we're always trying to which we always try to use to drive our research, and that's always looking to bridge translational gaps that we perceive are already existing in the literature. The first one I've already mentioned is with respect to the prefrontal cortex. Studies in rodents compared to humans, we need the monkey to bridge that gap. However, there's some other gaps we've also tried to bridge in the studies we've designed. The first is with respect to emotion per se, because how you measure emotion differs quite markedly between studies in rodents and studies in humans. In rodents, the primary measure of negative emotion tends to be a freezing response to a stimulus that they've learned is associated, say, with shock. Whereas in humans, even though they may use the same behavioral test, the same conditioned fear paradigm, they don't measure behavior, they don't measure freezing, because humans don't really freeze in that kind of situation. They measure autonomic activity, they measure skin conductance responses, or they measure heart rate. So they measure physiological responses. But ultimately, an emotional state is a product of the interaction between our physiological changes, the behavioral changes, and the subjectivity. That altogether produces that emotional state. So if you're going to measure emotion, I strongly believe you need to be able to measure more than one aspect of the emotional output. So in the marmosets, many of the studies I shall show you, what we do is we measure their cardiovascular activity alongside their behavior. And to do that, in a one-off surgical operation, we implant a probe into the descending aorta of the marmoset, which allows us subsequently to measure their behavior in freely moving animals in the test apparatus wirelessly. So we can measure both behavior and cardiovascular activity, again bridging that gap. And finally, the third gap we want to bridge isn't between rodents and humans, it's actually between rodents and humans and other studies in monkeys, because both rodents and humans, when studying negative emotion, tend to use Pavlovian conditioned paradigms, where animals learn and acquire a particular conditioned fear response to a stimulus that they've learned is associated with something nasty. Whereas some of the earlier studies done in the rhesus monkeys used paradigms such as innate fear, their innate fear responses to a snake, or anxiety responses, for example, to an unknown human, where you don't know the human's going to perhaps give you food or try and catch you, so there's an uncertainty and an anxiety associated with it. But in fact, we know to a certain extent that these kinds of different innate fears or anxiety may not completely work through the same circuits as these learnt fears. And therefore, in the marmosets, in fact, what we've designed is we don't only look at responses to snakes and human intruders, so we can translate our findings to what's already been gone forward in the rhesus monkeys, but we can also translate our findings into make sense of them in respect to rodent and human data, because we've developed fear conditioning paradigms in the marmosets. And the two areas we first decided to look at with respect to anxiety were the ventral prefrontal cortex, the orbitofrontal cortex, and the ventrolateral prefrontal cortex, because these regions have been shown to show altered structural and functional activity in depression, as well as a whole range of anxiety disorders. So they look like a good starting point. And these were areas that hadn't traditionally been looked at all in the rodent literature, in part because it's not clear rodent prefrontal cortex has a ventrolateral region, and up until more recently, no one had really looked at the orbitofrontal cortex with respect to rodents and fear conditioning. So the original paradigm we developed this Pavlovian paradigm in the marmosets, we use aversive loud noise as our unconditioned stimulus rather than foot shock. You're not going to shock the monkeys. But aversive loud noise has been used very successfully in human studies. And so animals are presented with one of two cues which come on for 20 seconds, two sounds, perhaps a tone and white noise. And after that 20 seconds, after one of them, the aversive loud noise, which is a punctate between 0.3, 0.5 of a second, 120 decibel sound. And they learn that one of the cues, say the sound, the white noise, is associated with aversive loud noise. And we compare their cardiovascular and behavioural changes within this CS periods compared to the 20-second baseline period. So within a single Pavlovian conditioning session, they'll receive up to 6 CS pluses, the ones that are associated with aversive loud noise, and 6 what we call CS minuses, which aren't associated with anything aversive. And this shows you the conditioned cardiovascular changes that you see. This is change from baseline. So during baseline, it's at 0. Then when the CS minus comes on, which predicts nothing, there is no change in the cardiovascular reactivity. This is heart rate. Whereas when the CS plus comes on, that ultimately predicts the aversive loud noise, you see this rise in heart rate. So this is your conditioned cardiovascular response. And alongside that, behaviourally, they show this enhanced vigilant scanning response during the CS plus, but not the CS minus. And this is published quite a long time ago now, and I'm not going to go into the details of this study, but just to tell you that what we found was that when we lesioned either the orbitofrontal cortex or the ventrolateral prefrontal cortex, and we did this using selective excitotoxins that literally destroy the cell bodies within these two distinct regions, we found that if the animals had already learnt this condition discrimination prior to surgery, post-surgery, the lesions had no impact on the overall expression of their conditioned fear responses if they'd learnt them prior to the lesion. What we did find, however, is that if we then change the relationship between the CS plus and the aversive loud noise, so things weren't quite as predictable anymore, whereas the control animals tended to show their conditioned cardiovascular and behavioural responses to follow and track the various changes that were going on in the environment, the lesioned animals didn't. They showed very rigid conditioned fear responses based on what they'd originally learnt, and they didn't really adapt them to changes in the environment. We then went on to look at the effects of these same lesions in the same animals on the two tests that have been traditionally used in rhesus monkey studies. Responding to a human intruder where an unknown person comes into the room and stares at them in one of the quadrants of their home cage for 2 minutes. That's enough to make them slightly anxious. Or we put a rubber snake into the home cage for a couple of minutes. And so this is the two paradigms. You get a whole repertoire of behaviours associated with these two tests. Obviously, depending on how anxious they are, they go towards the back of the cage. They may not move around so much. They show a bit of head bobbing. Some of the specific behaviours change depending on whether it's a human there or a snake. And they also make some calls. So what we did was, having made all these measurements, we threw them all into a principal component analysis. And that told us that there appeared to be two main components where certain behaviours tended to cluster with one another. And the one I want to focus on is the emotionality component, because the behaviours that clustered on this component were locomotion, whether they were still or not, how far they were from the front of the cage, so that if they were at the back of the cage and not moving much and doing quite a bit of head bobbing, that was a very high score on that component. And that's what suggested to us that basically reflected their overall emotionality. And so we looked at the effects of making these two lesions, the orbitofrontal and ventrolateral PFC lesion, on these measures. And what I'm showing you here is performance on the snake test on the y-axis and performance on the human intruder test on the x-axis. And all the grey dots are individual animals that we've tested in the colony. And this shows you the huge range of trait anxiety in these animals. So there are some animals that show really high anxiety responses to snakes and human intruders, and another range of animals that actually don't show much of an anxiety response at all. In fact, they're more interested as opposed to being scared. So this is the overall range in the colony. It just so happened that the animals in our lesion study shown here in blue before the lesion, were relatively low anxious. And there are reasons for that, if anyone's interested afterwards. But they were low anxious. However, having had that lesion of either the orbitofrontal cortex in orange or the ventrolateral prefrontal lesion in green, their anxiety scores, their emotionality measure on both the snake test and the human intruder test goes up. So there you can see it's going up on the snake test axis and it's going up along this way on the x-axis. So they're showing enhanced anxiety. But if you look at them in relation to the entire colony, they're not ridiculously anxious. They're not so super anxious that they look like no other animal ever existed. They're just showing more anxiety than they did previously. But clearly there are animals in the colony that are still much more anxious than they are, and they don't have, to our knowledge, a particular brain lesion. But they may well have dysregulation in some of these areas. So overall then, what I've shown you from this first study is that both lesions of this orbitofrontal cortex and lesions of this ventrolateral prefrontal cortex produce rather similar effects. Heightened fear conditioning, make it more rigid, heightened anxiety and heightened innate fear responses. But we know these two areas are not doing the same thing. These two areas are very distinct, have very distinct cognitive functions. So our next question was, what exactly are the distinct cognitive functions that are contributing to what ostensibly looks like a very similar syndrome of being more anxious and showing rather inflexible fear responses? So to do that, we moved on to a more sophisticated cognitive paradigm, and we chose to look at what we'd call a decision-making paradigm, but it's often called an approach avoidance task. So in this task, we initially trained marmosets to respond to two stimuli on the screen, which are associated with gaining access to reward. And this was the work of Hannah Clark in the laboratory. And responding on both of these stimuli give you reward on some kind of variable interval schedule. So what that means is that after a certain variable delay, the first response on the screen afterwards gives you reward. So what the animal should do on this task is respond relatively sustained and constantly, because it never quite knows when the next reward's coming, and also switch their responding between the two stimuli, because they'll maximize their responses by performing on both stimuli. And that's the reward-only condition. But occasionally, we introduce punishment, but we only introduce punishment on one side, in this case on the left. So on that particular session, which we only perhaps run once a week, they'll come in, they'll be responding away for reward, and suddenly a response on that side, punishment occurs. And the punishment is that aversive loud noise I've described already. So if they don't like the punishment, and they find it really aversive, they can avoid it by just doing all their responding on the other side. They won't get the maximum amount of reward, but they will still get quite a lot of reward by doing so. But in fact, what we found was our marmosets actually found the reward too nice. And so they carried on responding, even on the side where they got the punishment. And this is the sound that previously I've shown you produces quite strong Pavlovian conditioned fear responses. But nevertheless, as soon as you put it into a situation, it's relatively mild. And when you put it into a situation where now they're getting reward, you find that actually they'll carry on responding. But the question arises is what happens if we inactivate those two regions of prefrontal cortex? This time, rather than doing lesions, we've now moved on, and instead we cannulate these regions. There's the ventrolateral cannulation and the orbital cannulation, and we infuse down there a cocktail of GABA-A and GABA-B agonist drugs that act to excite the GABAergic inhibitory interneurons, which effectively dampens the activity in that area, but only temporarily. So on any one session, we might suddenly one day inactivate that area, and it will last for 20, 30 minutes, and then we can look at the effects of that inactivation temporarily on their performance in this task. There is no effects on overall responding for reward. Inactivating either of these regions makes no difference to the animal. But we did find an interesting effect when we look at the effect of inactivating either of these regions on this probe task, on this probe session. This first graph shows you what happens when we inactivate the ventrolateral prefrontal cortex. This shows you a bias of responding. So if you're responding at 100%, you're equally responding on both sides of the screen. You don't care. And this shows you the change from saline. So this shows you that anything above the line means that you're actually showing a bias away from the punishment. And this is different from the control, the saline condition, when we just injected saline, which has no effect. So what you can see, when we inactivate the ventrolateral prefrontal cortex, they show this strong bias away from the punished side, which they didn't do under normal saline conditions. So it was as if the ventrolateral prefrontal cortex had enhanced the punishment in some way, such that they wanted to avoid it a lot more now. In contrast, when we inactivated the orbitofrontal cortex, on the day that we did the inactivation, when they received punishment, there was absolutely no effect. The animals carried on responding normally, the same as saline. But what we found was this next day, and you're probably all wondering why the next day was there. This is why the next day is there on the graph, because on the next day, when there is no punishment, there is no infusion into the brain, this is just next day, but suddenly the animals with the orbital frontal inactivation from that day show a marked bias away from the punishment. But their OFC is not being inactivated that day, there is no punishment that day. So the only reason they must be avoiding the punished side from yesterday is because somehow they remember the punishment, and remembering that punishment, it must be quite a strong punishment memory to make them avoid it. So it appears that the OFC increases punishment avoidance, but not on the day when you've inactivated the OFC, on the next day. There's no effect of either of these manipulations on overall responding in the task. So our next question was, if they're somehow got this strong memory of punishment from the day before when you've inactivated the OFC, first of all, what's driving this punishment memory? Two obvious structures are the amygdala and the hippocampus, those subcortical structures that we know are critical for learning about punishment in the environment and showing expression of punishment. So Hannah went on and cannulated in the same animals the amygdala and the hippocampus. And then we asked the following question. We know that if we inactivate the OFC this day when they're receiving punishment, there's this enhanced punishment bias the next day. Where's that punishment bias coming from? Where's the memory coming from that drives that bias? If it's coming from the amygdala, if we inactivate the amygdala on that next day, we should block the bias. And that's exactly what we found. So that now, if we inactivate the amygdala on that next day, having inactivated the OFC the day before, during punishment, they no longer show the bias. They also no longer show the bias if you inactivate the hippocampus, and also they no longer show the bias if you disconnect the amygdala from the hippocampus. So in all, it would appear that the amygdala and hippocampus downstream are important circuit for expressing that strong punishment memory the next day. But what this really is interesting is it shows you that the orbitofrontal cortex, when it's normally online, even though it's not needed to avoid punishment on the day, nevertheless, it is learning about what's going on that day, and it's actually impacting on what's learned and what's laid down in structures such as, for example, the amygdala or the hippocampus. So it's affecting our overall perception of the punishment on that day, such that the following day, when we haven't had the OFC online, the amygdala potentially has got a stronger punishment memory than otherwise it should have. So the OFC appears to be modulating what we lay down in terms of our memories about punishment. Of course, we're now going on to do studies to allow us to explore that in more detail. For example, we want to now address the question, does the orbitofrontal cortex need to talk to the amygdala and or the hippocampus on the day of the punishment in order to influence the laying down of the memories? Is that memory being laid down differently in the amygdala or the hippocampus on the day? All we've shown so far is the amygdala and hippocampus is important for the expression of that memory the next day, not necessarily the actual where the memory is stored. So in summary then, what I've shown you from these two studies is that the orbitofrontal cortex has a rather distinct pattern of deficits on this task to the ventrolateral prefrontal cortex damage. So how do we interpret this? Well, if we think about what we know about the anterior orbitofrontal cortex in general, we've got some wonderful studies that have been done in both rodents from Jeffrey Schoenbaum's lab, from Betsy Murray, in monkeys, that tell us that the orbitofrontal cortex is very important for learning about and updating information about rewards and punishments in the environment, for learning to predict punishments and rewards, and particularly in updating that information as the environment constantly changes. If you can't do that, because you've inactivated the orbitofrontal cortex, what happens is that you become uncertain, because you can't quite predict what on earth's going on in this changing environment. If you become uncertain, that makes you more anxious, because being uncertain is anxiety-provoking. So we would argue that the reason you have general anxiety effects with damage to the orbitofrontal cortex is because you've knocked out your system that's good at tracking the changing punishment and reward in the environment, and that makes things uncertain and therefore makes you anxious. But what about the ventrolateral prefrontal cortex? We showed many years ago now that this structure, the ventrolateral region, was really important for an attentional control function. We showed that if you lesion this area, you impair the ability of an animal to switch their attention from one dimension of the world to another. So how do we think about that in terms of anxiety? Why should it induce anxiety? Well, one hypothesis that we propose is the following. When that animal is in the apparatus receiving its reward, if suddenly aversive loud noise happens, that's a really salient event. Nasty things are salient. So you suddenly find your attention dragging to that aversive noise. But in this context of decision-making task, you need to then draw yourself back to the context, the reward you're receiving, to ultimately make a decision. Do I want to carry on responding for reward regardless of this punishment, or actually is this punishment so bad and the reward not that good that I'm actually going to start trying to avoid it? In order to do that, you have to switch your attention back from that very salient stimulus. And I would argue that one reason why they may have a problem in doing that is because the ventrolateral prefrontal cortex is important for switching attention. And if you can't switch your attention because you've inactivated that very region that normally allows you to do it, then you find yourself stuck paying attention to this aversive loud noise, and you can't switch back from it. And any of us, if we spend all our time paying attention to something nasty, we will become anxious. So one proposal then is these two regions are producing anxiety, but for two very different reasons. One, because they have a problem switching attention. The other, because they have a problem updating and tracking punishment and reward in the environment. Which means if you take a patient with anxiety, they may have a disruption in one or other of these circuits, but you're going to treat them differently. There may be different neurochemical drugs that we could use to differentially affect activity in either of these circuits. And there obviously are probably cognitive behavioral therapists that you'd use differently to help someone who has a problem updating versus someone who has a problem shifting attention. So this is why it's really critically important to understand the underlying neural circuits for what produces what ostensibly looks like the same overall effect of increased anxiety. Finally, we went on to ask one more question with this circuit, and that was, although these two areas are performing different functions, do they act and converge on the same downstream emotional circuits, or do they operate in parallel? And to answer that question, we turn to micropet and to fluorodeoxyglucose. Fluorodeoxyglucose gets taken up by cells in the brain that are very active. But deoxyglucose can't be broken down to form energy. And so it stays in the cell and accumulates. And so it's a proxy marker of activity in the brain. And it was used a lot before functional imaging came on the scene in human studies. And it's still been used a lot, for example, in studies of depression. So we use fluorodeoxyglucose to look to see what changes are going on downstream in the brain when we make these manipulations in the orbital frontal and ventrolateral prefrontal cortex. So the design is complicated because we wanted to maximize the power of the design. So we had a unilateral lesion model because we know that primarily orbital frontal cortex on one side talks to downstream structures on the same side. There's very little crossover, and we've got that from our own marmoset tracing studies. So we did unilateral lesions of either the orbital frontal cortex or the ventrolateral prefrontal cortex, so we've gone back to lesions now, it's cell body lesions, in separate groups of animals. After they would receive the lesion, we waited 3 months, and then we performed 3 scans. The first scan, they were given the FDG, and then for half an hour, you put them into a behavioural test situation, and the situation we put them in was fear induction. They were put into the test apparatus where they saw a rubber snake and the lights went out, and that made them rather anxious. Half an hour later, at the end of that period, you anaesthetise the animal, put them in the PET scanner, and look to see which brain areas were most active during that period, as measured by the uptake of FDG. We then waited 10 days. During that period, we habituated them to that test apparatus. So they went in the test apparatus every day after that for half an hour, but every other day there was never darkness, there was never a snake. They got used to it, and actually it was a relatively safe environment. And then after that 10 days, we gave them the FDG bolus again. They went back into that now safe environment, and then half an hour later, nothing happened in that environment. The light was on. They got a bit bored, probably fell asleep a little bit. But then at the end of that period, they were scanned, and we looked at activity again. And then finally, again, to help strengthen the power of our design, we did a replication of the fear induction, where we now put them in a different apparatus after the 3rd FDG injection. And this time there was a snake again, and there was darkness. And we looked to see what areas of the brain, first of all, on the intact side, were differentially sensitive to these 2 conditions, fear versus safety. And, not surprisingly, the amygdala lit up. So what this means is the activity here, there was FDG uptake in this area far greater in the fear condition, or 2 fear conditions, compared to the safety condition. So if you like, the amygdala knew whether it was in a fearful condition or a safety condition, based on the amount of activity that the area was showing and the amount of FDG it had taken up. That's on the intact side. But what about the lesions hemisphere, where the amygdala is perfectly intact, but there is now, in some animals, no orbitofrontal cortex, and in other animals, no ventrolateral prefrontal cortex? If you compare fear minus safety in those conditions, there is no difference in FDG uptake in the amygdala between fear and safety on the lesioned hemisphere. But if you look at exactly what the levels were, you find that the reason there is no difference is because FDG uptake during safety was as great as it was during the fear. So the amygdala was unable to know the difference between fear and safety. It assumed it was all fearful. That's either in an animal which had no orbitofrontal cortex on that side, or in an animal which had no ventrolateral prefrontal cortex. So in both cases, you see this change in activity in the amygdala. And this just shows you the story in more detail, because in fact it's not just in the amygdala. The area we found that shows this differential activity to fear versus safety included the dorsal amygdala, but it also went out into the anterior insula. And that's on the intact side. And like I said, that difference was not seen on the lesion side. And if you look specifically, that's the amygdala insula cluster that we got from the voxel-based analysis, showing more FDG uptake during fear, less in safety and back up in fear. If you look at purely the amygdala component of that cluster, you see the same pattern. If you look at the insular component of that cluster, you see the same pattern in the intact side. And like I said, in the lesion side, I haven't got the graph to show you, but it's a flat line across because it's high in all the conditions. So in summary then, it appears these 2 separate pathways nevertheless are converging on downstream structures, including the amygdala and insula, in order to regulate our emotional responses normally to changes in the context of the environment. But without those areas, of course, the amygdala suddenly becomes blind in a way to the various contextual situations you're in, where you are normally expected to behave differently. I'm now going to change tack a little bit and move on to the other circuit we've been looking at with respect to anhedonia. Anhedonia is the loss of pleasure. And we know from studies in depression that one of the areas that shows marked changes in activity in depressed subjects is this region, the ventromedial prefrontal cortex. I'm using Michael Petridis' cytoarchitectonic analysis of the area. And a variety of changes have been shown. Helen Mayberg has shown altered activity, in particular increased activity in these caudal subgenial regions, area 25, but it's not always that caudal. It can be quite a lot more anterior than that. Also, there have been both inhibitory and excitatory changes associated with these more rostral perigenial regions, perhaps area 32. And finally, there have been changes reported in depressed subjects in the dorsal anterior cingulate. And in many of these cases, these changes seen in depression are reversed following successful treatment, but not reversed following unsuccessful treatment. But like I said before, we have no idea whether these changes are compensatory or causal, and also what symptom are they responsible for. So we, in the first instance, have cannulated and targeted area 25 and area 32 in the marmoset, and look to see if we activate or inactivate these areas, what are the effects on anhedonia. And again, we are looking to bridge translational gaps in the study of anhedonia, because in anhedonia in humans, the primary measure is a questionnaire. To find out how anhedonic or hedonic you're feeling, you get asked a whole load of questions about your state when you're experiencing various rewards. That's the measure in the clinic. In rodents, the measure that's been used a lot for doing translational studies is sucrose consumption or sucrose preference. They ask the rat to choose between a bottle of water and a bottle of sucrose. And as you might imagine, rats like sucrose, and if they show a strong preference to the sucrose. But in various anhedonic models, that preference decreases, it's gone. They seem to drink water as much as sucrose, suggesting they're anhedonic. But there's a big problem here, because if you look at depressed subjects, many of them don't have what you might call consummatory anhedonia. If you give them nice wine or you give them nice food, they actually think it's quite nice. What seems to be missing is either the ability to anticipate the nice food or the nice wine, or the ability to work for it. So that's the gap. But we know actually from experimental studies in rodents and primates that reward processing is far more complicated than that, and there are anticipatory components to reward. There are these motivational components, how hard you're prepared to work for reward, and of course there are consummatory components. So when we're trying to study these in meaningful, looking at circuits, we need to be able to take into account the different forms. So in the marmoset, so far, we've chosen a paradigm that allows us to look at both anticipatory and consummatory aspects of reward processing. So we're back to our simple Pavlovian conditioning paradigm, but this time for appetitive reward. And so animals are presented with either a CS plus, such as a sound, a tone, that after 20 seconds you get access to this big box of your favourite food, which is a box of marshmallow, or they get a CS presented which is associated with an empty food box. And again, they get a series of these in the session. And again, we can look at cardiovascular changes and behavioural changes. And just as I showed you for the negative conditioning paradigm, this time it's blood pressure, changes from baseline. Obviously, at baseline, it's 0. If the CS minus is presented, which doesn't predict getting access to food, nothing happens. Whereas if the CS plus is presented, which the animals learn predicts access to getting to the marshmallows, it rises. And then it rises even more when they're consuming the marshmallow. And there's also nice behavioural changes. So what I should point out then, here we have in the one test, both an anticipatory phase and a consummatory phase. And then behaviourally, anticipation-wise, you can measure their kind of orienting head responses they make to the CS that predicts getting access to food, but not the CS minus, which doesn't predict food. And consummatory-wise, you can measure how much they consume of the marshmallow when they get it, and also how long it takes them to actually start eating it, measures of consummatory arousal. And so we did 2 manipulations. We first activated or inactivated the area 32, both of which have been suggested to occur in certain forms of depression. and there were no effects. What this shows you is either the blood pressure change to the CS plus versus the CS minus, so that's our good, strong cardiovascular arousal response, and that's our strong behavioral arousal response to the CS plus, anticipation. If we inactivate area 32 using these GABAergic agonists, no effect. They still show strong cardiovascular and behavioral arousal effects in anticipation of reward. If we activate this region, and we activated it using a mixture of a GABA-B antagonist and an mGluR2/3 agonist, which basically results in increasing the amount of glutamate that's released, so it makes the area more active. This also had no impact. So the changes that we're seeing in depression, that may be either reductions in activity in this region or activations, don't produce a basic effect on either consummatory or anticipatory arousal. But a very different story emerges when we overactivate area 25. When we overactivate area 25, this again is our cardiovascular arousal response to anticipating reward. But when we activate this region, we make it more active, which is what has been suggested very often you see in depressed subjects. We completely blunt the cardiovascular arousal response. And we blunt it using two different methods to cause overactivity. The one I've already described, but the other one, we use dihydrochinic acid, which is blocks the uptake of glutamate in astrocytes. So what it means is there's more glutamate sloshing about. And whether there's more glutamate sloshing about because we've blocked the reuptake of it, or there's more being released, either way, we blunt the reward anticipatory arousal responses. So they don't seem to be that bothered about getting access to the food. And the same effect on the behavioural measure. We blunt it. However, if you look at consummatory measures, both the cardiovascular and behavioural of consummatory measures, they're intact. The animal eats as much, he's as quick to get the food, he just doesn't seem to show the excitement in anticipation of it. We've subsequently, and this is just trying to show off, we finally got dreads to work in the marmoset. So I'm showing off that we've used an excitatory dread, which is where dreads are what are known as designer receptors exclusively activated by designer drugs. And they are engineered G-protein coupled receptor that, when activated by an otherwise inert drug, such as clozapine N-oxide, CNO, actually activates these receptors, these designer receptors. And in this case, we're using the HM3DQ receptor, which is excitatory. So if you excite this receptor, you increase burst firing in the area. So it's yet another method of overactivating area 25. And when we use this, we get the exact same effects as we got with the other two methods. Overactivating area 25 bluntens the anticipatory arousal, both cardiovascular and behavioural, at the 6 and 10 mg per kg dose. We don't see it at the 3, but this fits in with what we know about these doses in other primate studies. But interestingly, again, no effect on consumption. So we've replicated the effect a third time using dreads. But we also, being conscious of the fact that we hadn't compared consumption in the way they do in rats, this sucrose preference test, we've actually looked at sucrose preference in the marmosets. So in the marmosets, we also give them a bottle of water and a bottle of sucrose. We let them choose which one they'd prefer, and they can spend up to an hour, 2 hours choosing between these two. And normally, marmosets, just like the rodents, will choose sucrose over the water. If you activate area 25, which bluntens their anticipatory arousal responses, it has no effect on their sucrose preference. They appear to be enjoying sucrose as much as any other animal. So they haven't got a consummatory anhedonia. What we blocked is their anticipatory anhedonia. They aren't anticipating something nice anymore. But to show that our test is working in the marmoset, given that we've never really done this before, we use naloxone, which is an opiate antagonist, and show that in fact it does block the effect. So finally, we wanted to ask the question, can we reverse this anhedonic effect? And we chose to look at the effects of ketamine. So as I mentioned at the beginning, depression Some patients do very well on certain antidepressants, such as the SSRIs, but some don't. And the ones that don't are what we call the treatment-resistant subjects, who don't seem to do well on any of the typical pharmacotherapies or behavioural therapies. And it's been shown that many of these patients do seem to show quite strong anhedonic deficits, and they seem to be particularly treatment-resistant to the SSRIs. But recently, ketamine has been suggested that it can actually act as an antidepressant and have quite strong effects quite quickly, although as a one-off dose, they last only sort of for a month or so. So ketamine may be effective at treating some of these patients who don't respond to SSRIs. So we just asked the question, does ketamine have any impact on our animals, our anhedonic effect produced by the overactivity in area 25? So this is the last study I will show you. We injected ketamine in these same animals, and then either 4 hours afterwards, or 1 day, 7 days, or 21 days, we then injected dihydrochinic acid, so we overactivated area 25 as normal. And of course, if we overactivate 25, we're expecting to see a blunting of the anticipatory arousal response. We're looking to see anhedonia. But now we're asking the question, can we block that anhedonic effect by giving this single dose of ketamine? And the short answer was yes. Ketamine has no impact on any of these measures on its own. But if we look at the impact of DHK after this single dose of ketamine, at 4 hours after ketamine, the DHK, the overactivity of area 25, blunted the anticipatory arousal, produced the anhedonic effect. However, at 1 day and 7 days, the DHK was no longer able to produce the anhedonic effect, but by 3 weeks afterwards, it produced it again. So it appears that ketamine does reverse, the one-off dose of ketamine does reverse this anhedonic effect produced by the dihydrochinic acid. So I'm going to leave it there, and I'm going to not show you the last studies. So just to summarize then, what I've shown you today so far is that area 25, what I didn't show you was some of the imaging studies that implicate these other two regions in this effect, seems to be quite important, and when overactivated can produce appetitive anhedonia, anticipatory anhedonia, selectively for anticipation and not consummatory, which means that this looks far similar to the anhedonia you see in many depressed subjects. And we've shown you how ketamine can reverse this effect. What we obviously want to go on and do is look to see whether SSRIs can also reverse this effect or not. So we can begin to start dissecting out the neurochemical modulation of these circuits. And likewise, I've also shown you the role of different parts of the prefrontal cortex in regulating downstream negative emotional systems, which can lead to anxiety. And again, what we're currently doing is looking to see whether ketamine, the glutamatergic antagonist, or SSRIs, how selective they are for manipulating or modulating these circuits. And so ultimately, what we're hoping that's going to be able to do is we're beginning to fractionate out the pathways that underlie different forms of anxiety and different forms of anhedonia, and by determining how these are modulated by these distinct neurochemical systems, we might be getting one step closer to being able to provide individualized treatment strategies. And I shall leave it there, and there to thank the colleagues, in particular Dr. Hannah Clark and Mr. Laith Alexander, who's done most of the anhedonia work, who's a PhD student in the lab. Thank you.